some weird finding, and Francie would be sitting there eating lunch and say, oh yeah, we see that all the time. And my PI would just be like, oh, I didn't realize that. Or she'd always back us up. It was great. And also a trailblazer for me because both of us ended up leaving New York. Francie came out to UCLA to do her postdoc with Pam Johnson, and then now is an assistant professor at Cal State Pomona and studies Trichomonas vaginalis and also potentially other trichomonads in other species, hopefully, and the immune system. And she's going to tell us about the human immune response to Trichomonas. So thank you, Francie, for joining us. Sorry it's not in person, but I'm really glad you could speak to us.
Okay, no problem. This is a fun break during the pandemic. So I'm going to share my screen with my slides on it, and please feel free to unmute yourselves and interrupt me with any questions that you may have, or stop me if something's not clear. And so I will get started. Okay, so share screen. Okay, so can everyone hear me okay and see this laser pointer?
Yes, we see your laser pointer.
All right, I can figure out where to put this so that I can see all my slides. Maybe I'll just make it smaller. Okay. Okay, so today I'm going to talk about how the parasite Trichomonas vaginalis interacts with different human leukocytes. So this is a picture of the parasite, Trichomonas vaginalis, and this is a picture of our campus. So I'm at Cal Poly Pomona, so we're in the CSU system, and there's 2 polytechnic campuses within the CSU system, so we're one of those. And we're in Pomona, which is about 30 minutes east of LA. So I just started there 2 years ago. So a lot of the work I'm going to be talking about today was work that I did during my postdoc in Patricia Johnson's lab at UCLA. Okay. So Trichomonas vaginalis, which I'll abbreviate as TV on a lot of the slides, is a unicellular flagellated protozoan parasite. And it's exclusively an extracellular pathogen, so it doesn't actually enter any of our cells. And as you can see here in this beautiful EM taken by one of my colleagues in Brazil, you can see that the parasite kind of has 2 morphologies. So here is shown the free swimming form. So you can see it has 5 flagella and it has this trophozoid form, so it can swim. And upon encountering a host cell, so here shown here, this is a vaginal epithelial cell. The parasite can adopt an amoeboid morphology so that it can cling to the surface of those cells. So it attaches to epithelial cells in the urogenital tract. And oftentimes the parasite also subsequently lyses these cells, and it's thought that feeding off of those contents is a mechanism by which the parasite gets nutrients. And it's also thought to be a mechanism of pathogenesis of the parasite, the lysis of these urogenital epithelial cells, like vaginal and cervical epithelial cells. So the field of Trichomonas biology kind of had its 15 minutes of fame in 2007 when the genome was sequenced. And so these beautiful pictures made them onto the cover of Science. And so the sequencing of the genome has enabled us to do a lot more molecular biology with the parasite and to get at more mechanistic things regarding its pathogenesis. Okay, so with this audience, probably most of you are familiar with the fact that Trichomonas causes trichomoniasis, which is one of the most common sexually transmitted infections that we have in the US and worldwide. So it's the most common non-viral STI. So if HPV and HSV2 are the 2 most common STIs that we have. So trichomoniasis is number 3. So this is more common than all of the bacterial STIs that we more commonly hear about, like chlamydia, gonorrhea, and syphilis. It's more common than all of those combined. So it's a very prevalent STI. So in the US, it has a prevalence of about 3.7 million. And annually, it's estimated that there are between 1 and 10 million new cases. It's also very prevalent across the globe. And importantly, this is yet another contributor to health disparities that we have in the US and worldwide. So it really disproportionately affects underserved communities, and in particular is about 10-fold greater incidence in Black women compared to other women in the US. And because of its high prevalence and the fact that there's really not that much research done on this parasite, it's actually classified as a neglected parasitic infection in the United States. Okay, so symptomatic cases with this parasite can cause vaginitis or prostatitis, so inflammation of the infected tissues. But importantly, and it can be treated with the 5 nitroimidazole classes of antibiotics. So those are antibiotics that exploit the anaerobic metabolism of the parasite. But alarmingly, up to 10% of cases are now reported to be resistant to antibiotics in studies of different isolates across the globe. So we do have a problem with drug resistance rising to this parasite. So the backup to metronidazole is tinidazole, but there are emerging resistant cases to tinidazole as well. And another reason that trichomoniasis is a problem from a public health standpoint is because so many of the cases are asymptomatic. So this really facilitates spread and can lead to infected folks not seeking treatment. And so as a result, untreated or asymptomatic infections can lead to some complications. So trichomoniasis has been associated with both male and female infertility. It's been associated with preterm labor, so this can cause low weight infant births. And again, this is something that disproportionately affects Black women. And it's also associated with pelvic inflammatory disease. And it's also been associated with an increased susceptibility to and transmission of HIV. So it's thought that Trichomonas infected women are 2 to 3 times more likely to contract HIV. So this can contribute to spread in communities and areas where that's endemic. And also to increase the incidence of some invasive cervical cancers. So we know that cervical cancers are caused by HPV, but in women that are already HPV positive that have neoplasms or neoplasias, having a Trichomonas infection at the same time can increase the chances that those become malignant. So, it's interesting from an immunological standpoint because we think that a lot of these complications or adverse sequelae or effects associated with trichomoniasis are really associated with inflammation as a result of the parasite. And so I was really interested. I finished my PhD in T-cell immunology in 2013, and I wanted to get into a more understudied area. And so I was really interested in characterizing the immune response to this parasite when I started my postdoc. So one of the reasons that the immune response to this parasite is so under-characterized is because we don't have great mouse models to do pathogenesis studies. So the establishment of mouse models is really challenging. So this is a human-specific parasite. To infect mice intravaginally, we have to use really high levels of the parasite, and it's really difficult to get any parasites to remain in the vagina and sustain an infection. And a lot of that is thought to be because of differences in pH between mouse and human vaginas, and also differences in the microbiomes. So trichomoniasis, as I will mention a little bit later in the talk, seems to prefer different vaginal microbiomes to others. So this could also be a reason. So what we do know about the immune response to it is gathered from clinical data. So we know that partner reinfection is common in trichomoniasis. And so women can present, after they've been treated with metronidazole, they can present again with the same strain of Trichomonas weeks later, indicating that they contracted it back from the partner. And so this indicates that we may not get really good formation of immunological memory in most infections. And another thing we know is that neutrophils are recruited to the site of infection. And so we know this because one of the clinical hallmarks of a trichomoniasis infection is this white frothy discharge. So this is shown coming from a cervix. And if you take that and you put it on a microscope slide, you can see that a vast majority of the cells there are neutrophils. So we know that they are recruited to the site of Trichomonas infection. So some overarching questions that I had during my postdoc and that I'll touch on today are whether these cells that are abundantly recruited to the site of infection actually kill the parasite, whether or not the parasite evades host immunity. And I think we have a sense that the answer is yes, because we know that these infections persist and that they can recur. But how does this happen? How does the parasite evade host immunity? And also, how does the parasite cause disease? So one of the first things I did, so getting at the first question of whether neutrophils can actually kill the parasite, one of the first things I did is just set up competition experiments between neutrophils isolated from peripheral blood of healthy donors and parasites and Trichomonas. So I differentially labeled them in 2 different dyes so that I'd be able to tell them apart. And then we co-cultured them together in a cell culture dish for 2 hours. And then we use flow cytometry to count the survivors. So count number of red cells, count number of green cells, and see who survived when you put them together in this competition experiment. And what we found is that the neutrophils won. So I never saw instances where the parasites killed the neutrophils. So even though Trichomonas has this lytic behavior toward vaginal and prostate epithelial cells, it doesn't appear to be killing neutrophils. And what we found is that, in fact, neutrophils were able to kill the parasite. So we set up this experiment at various different cell-to-cell ratios. So here you can see at a ratio of 1 neutrophil to 1 parasite, we achieve about 30 or 40% killing. And then as we increase the number of neutrophils, we see more efficient killing and can get complete killing of the parasite at higher neutrophil numbers. And so we think this is physiologically relevant because we know that neutrophils are abundantly recruited to infection sites and that they attack in aggregate in swarms. And actually, if you look at samples of vaginal swabs in trichomoniasis patients, estimates are that neutrophils outnumber the trichomonads there by 100 to 1. So we think that these ratios are, it's reasonable to think that they would be physiologically relevant and that this killing is actually quite efficient. We also did this with neutrophils from many different donors' blood to confirm that this was reproducible and that this wasn't an anomaly that we were seeing in just a couple of folks. So it does seem that across the board, neutrophils from many different individuals have the ability to kill this parasite. So our next question was, how are neutrophils killing Trichomonas? So neutrophils have 3 main mechanisms that they use to kill pathogens. So the first mechanism is phagocytosis, so engulfment of pathogens. And subsequent to phagocytosis, the phagosome can fuse with a lysosome to form the phagolysosome, where pathogens are degraded. Or in the case of neutrophils, neutrophils have these toxic granules, which contain things like pore-forming toxins and reactive oxygen species and proteases and lipases. So in neutrophils, phagosomes can also fuse with toxic granules to intoxicate pathogens in that way. If pathogens are too large or motile to engulf, oftentimes neutrophils can exocytose the contents of these granules into the extracellular environment to intoxicate pathogens that way. And another way that neutrophils can kill pathogens that are on the outside that they don't engulf or can't engulf is through this more recently described process called netosis. So NET stands for neutrophil extracellular trap. And this is actually when neutrophils extrude the contents of their nucleus and the DNA unravels and forms this web or this net-like structure that can ensnare pathogens. And then the pathogens can subsequently be killed by those toxic granule components that also come out with the net. Or a lot of times it just traps the pathogen there so that phagocytes can subsequently come and clear the area. So we wanted to know which of these mechanisms neutrophils use against Trichomonas. And so we decided to take a process of elimination approach where we blocked one pathway at a time and then asked if the neutrophils could still kill the parasites. So which of these mechanisms do they use? So our first hypothesis was that neutrophils are killing Trichomonas using netosis. And so to test this hypothesis, we utilized DNase, which is an enzyme that degrades DNA. And a lot of researchers have shown that adding DNase to your cell culture degrades nets and can allow survival of pathogens that would otherwise be killed with nets. So we used the same co-culture system that I showed in a previous slide, where we label parasites in red, neutrophils in green, let them duke it out, and then count survivors. Except this time we added DNase into the wells. And we did confirm that that DNase was able to degrade any nets that the neutrophils formed. However, we found that this made no difference in the ability of the neutrophils to kill the parasites at any of the ratios that we tested. So we concluded that neutrophil killing of Trichomonas is not likely to be mediated by netosis. Okay, so our next hypothesis is that neutrophils were killing the parasites using the extracellular degranulation and intoxicating the pathogens with these more soluble mediators. And so one of the ways that we tested this was to ask whether or not the killing required cell-to-cell contact. And so we utilized transwell plates. So these are plates where you can put two cell types together where they share continuous media, but you can put one cell type up in the top chamber and one cell type down in the bottom chamber. And the two chambers are separated by a porous membrane through which cells can't pass. So the cells won't be able to come into contact with each other, but all of the components of the toxic granules can get through those pores. So we set up the cytolysis assay in two different ways, either having the parasites and the neutrophils together in the same chamber or separating them by putting the parasites in the top chamber. And when we did this, we found that only when the neutrophils and the parasites were cultured together were the neutrophils able to kill the parasites, and that when the parasites were separated up into that top chamber, we got no killing. So we thought maybe this isn't a totally fair experiment, because maybe the neutrophils need to come into contact with the parasites to become activated in order to degranulate. So we added some different conditions where we stimulated the neutrophils in the bottom well, either with Trichomonas itself or with PMA, which is an artificial stimulus that induces neutrophils to degranulate. And even though in both of these conditions we did confirm that the neutrophils did degranulate, we still never saw any killing of the parasites in the top wells. So this indicated that neutrophil killing of Trichomonas was really strictly contact dependent. And so we thought that it was unlikely to be mediated by exocytosis of those toxic granules into the milieu. So that kind of ruled out the second hypothesis and left us with phagocytosis. So to test whether the neutrophils killed the parasites using phagocytosis, we added phagocytosis inhibitors to our assay. So we pre-incubated the neutrophils with either cytochalasin D, which is an actin polymerization inhibitor. So we know that neutrophils need to use actin polymerization in order to form the pseudopods and engulf. So there needs to be cytoskeletal movement. And wortmannin, which is a PI3 kinase signaling inhibitor, and PI3 kinase signaling is also required for phagocytosis. So in the presence of either of these inhibitors, now we saw a robust inhibition of the neutrophils to kill the parasites at all of the ratios that we tested. So we concluded that engulfment was necessary for the neutrophils to kill the parasites. Okay, so as a recap, the best hypothesis that we have now is that phagocytosis is the most likely mechanism that neutrophils use to kill Trichomonas. So our next question was, how do neutrophils phagocytose Trichomonas? And in this cartoon that I've been showing here, this appears to be like a bacillus bacteria. And so these are about 0.5 to 1 micron in size. And neutrophils can vary a bit in size, but in the blood, they're between 8 and 9 micron in average diameter. But Trichomonas is actually a little bigger than this. So Trichomonas are generally 10 to 15 micron in diameter. So we were a little complex about how this cell was eating something that was bigger than it. And so we wanted to get a look at what the neutrophils looked like after they had killed the parasite, so that we could ask whether this neutrophil can actually get all the way around this big parasite. And so to do this, we used imaging flow cytometry. So this is, you may have one of these at UCSD. At the time I was doing these experiments 5 or so years ago, this was kind of novel and exciting. But this is the traditional flow cytometer that, in addition to having lasers, also has a a microscope inside. So as the samples pass through the inquisition point, you take a photo of each cell. And that gathers bright field information as well as in several fluorescence channels. So on your FACS plots, you can actually gate on populations of cells and see what they look like, or you can click on individual dots on your FACS plots and see what those cells look like. So we did our same cytolysis assay that we'd been doing, where we co-culture the two cell types, except now instead of analyzing with traditional flow cytometry, we analyze them through imaging flow cytometry. And when we did this, what we saw, so again, we have our parasites labeled up in red, our neutrophils labeled up in green. And in the cells from the co-cultures, after they had been allowed to duke it out for 2 hours, what we saw was actually green cells, so neutrophils that look like they have spots of parasites inside of them. So this was consistent with a engulfment-dependent killing. But these spots are much smaller than the size of the parasite itself. So we thought that the most likely explanation for this is that the neutrophils are phagocytosing the parasites whole and then subsequently degrading them up into smaller pieces once the parasite's in a phagolysosome, or once that phagosome has fused with toxic granules. And so to test this hypothesis, we fed neutrophils some pre-killed parasites. So we have dead intact parasites that we killed with heat inactivation. And when we did this, we saw something very different. So now we did see, I can't really point to this because my Zoom window is in the way, but now we see what does appear to be neutrophil cells that were able to get around the parasite. And even though we continued to culture these for hours after, they never subsequently degrade the parasite into these smaller pieces. So this argues against the hypothesis that rather than phagocytosing the parasite whole and then subsequently degrading it into smaller pieces, that maybe these pieces of the parasite are taken by the neutrophil while the parasite is still alive. So this suggested that maybe bites of the parasite are taken before the parasite dies. And so this is consistent with a phenomenon called trogocytosis. So if phagocytosis means to eat or to eat whole, so trogo comes from the Greek word to nibble or to gnaw. So this is a process that's been described in several scenarios, but in 2014, there was a really nice Nature paper where Katie Ralston, who's now at UC Davis, showed that the amoeba, Entamoeba histolytica, actually kills host cells using trogocytosis. So instead of engulfing the host cells whole, it can actually nibble on the cells until they die. So take piecemeal bites. So there's been some other really interesting functions attributed to this cellular process in host pathogen interactions. So for example, in 2016, trogocytosis between macrophages in mice was shown to be able to spread intracellular bacteria, like Francisella tularensis. And in 2017 and 2018, neutrophils were shown to be able to trogocytose cancer cells. However, trogocytosis had never been shown as a way for immune cells to kill a pathogen, so we thought that would be really interesting if this was the mechanism at play. So in order to test this, we had to do live imaging, so we needed to watch it in action. And so we devised a live imaging strategy where we have, so these are the Trichomonas parasites, they're surface labeled up in green, and then we co-culture them with neutrophils. And in this experiment, our neutrophils are not labeled, so we just visualize them in bright field. But we added propydium iodide to the media. And so propydium iodide is a nucleic acid stain. So it stains DNA and RNA, but it can't permeate intact cell membranes. So this is a good way of tracking in real time when your cell's alive and when it's dead. So if the cell turns red, then that is an indication that the membrane's been breached and that the cell has died. So we can see at early time points during this experiment, and these were really fun experiments to set up, but challenging because both cell types are motile. And so you're trying to keep a focus on what's going on as the parasite's swimming around and the neutrophils are following it. But we managed to get some good videos. And so here you can see these 3 neutrophils have approached this parasite. And after about 30 or 40 seconds, you can start to see pieces of the parasite's membrane or little green blebs or bites present over in where the neutrophil cell bodies are. And if you just look at fluorescence only, you can see that these are discrete and separated from the main plasma membrane of the parasite. so consistent with neutrophils nibbling on the parasite. After a few more minutes, you see an accumulation of bites, and then several minutes later, you see that the parasite has died. So we never saw a propidium iodide signal inside the parasite until there had been an accumulation of a lot of bites in the neutrophils. So we did this with many different donors' neutrophils and analyzed many different videos. And we find that on average, it took about 7 minutes between when the neutrophils first start nibbling on the parasite membrane to when the parasite actually dies. So again, this argues against the hypothesis that the neutrophils are killing the parasite first and then eating up the pieces, and instead suggests that they're nibbling on it prior to the death. And so in fact, those nibbles could be causing the death of the parasite. So we wanted to get a better look at this using a different imaging technique. So we did 4D super resolution confocal live imaging. So this was done at the California Nanosystems Institute, which is housed on the UCLA campus. And so in this experiment, again, we have our parasite surface labeled up in green, and we have the neutrophils now labeled in a cell tracker to label their cytosol. And so you can see here, these 4 neutrophils have approached this parasite in our surrounding. Is it going to let me show the video with this pointer? Okay. So you can see right here, this neutrophil is coming and pinching off a piece of the parasite. And by the final time point of this video, so the video is on a loop, but by the final time point of the video, you can see that that piece of parasite is no longer visible. So we asked if that's because it's actually inside the neutrophil cell body at this point. And so we took a computational Z slice through the image, and we saw that indeed there is this little green fragment or bite of parasite material inside the neutrophil, and that it's discrete from the main cell body of the parasite. So this is consistent with the neutrophils nibbling on and internalizing pieces of the parasites at very early time points in their interaction. So this is shown at a bit of a later time point when a couple more bites have accumulated, and we can take, again, a cross section through those cells and see that indeed these bites do accumulate in the neutrophils when they encounter the parasite. So this is consistent with the process of trogocytosis. Okay, so as a recap, neutrophil killing of Trichomonas is blocked using engulfment inhibitors. However, they don't eat the live parasites whole. Instead, they appear to internalize bites of the parasite prior to and until the parasite's death. And these observations are consistent with the cellular process of killing by trogocytosis. So our next question, having made this observation, is what's the mechanism of this process? And so 2 more specific questions we have are, what molecules are mediating the contact between the 2 cells? And also, what enzymes could be mediating the nibbling, could be mediating the degradation of the parasite surface into these bites? And so one thing I haven't told you yet, that if anyone here has ever worked with neutrophils may have already figured out, is that in all of the experiments I've shown you so far, they were always done in the presence of 10% human serum. So, and in fact, many functions that neutrophils use to kill pathogens are dependent on human serum. So if we don't put any serum into our culture, we got no killing, but in the presence of human serum, we get killing of the parasites. And so this indicated to us that opsonins were involved in this process. So opsonins are serum proteins like complement and antibodies that coat the surface of pathogens and can help cross-link the pathogen to immune cells that have receptors for those opsonins, like FC receptors and complement receptors. So making this observation, our hypothesis is that the parasite needs to be opsonized in order to be trogocytosed. And so to test this hypothesis, the first thing we did was just to ask whether our parasites were being opsonized by the serum in our experiments. And so to do this, we devised staining schemes where we incubated the parasites with human serum, and then we came with an anti-human IgG. And in fact, the parasites do light up, so indicating that their surfaces were coated by a human antibody. And this is actually not surprising. So to do these experiments, we reserved a big batch of serum from Corning, which is pooled from 100 males. And so given what I've told you about the prevalence of Trichomonas, it's not surprising that there were some Trichomonas positive antibodies within that serum. So that's good for us because it helps us do our experiments if we have antibodies bind the parasite in there. And we also saw that, so the major complement factor that's responsible for opsonizing pathogens is called IC3B. And so we also found that the parasites were indeed coated with IC3B, because when we incubated the parasites with serum and then came with an anti-IC3B antibody, the parasites lit up. So we know that they get opsonized. So we want to now test to see whether the receptors for IC3B on the neutrophils and also receptors for antibodies on neutrophils are essential for this trogocytic process. So this is one of the things that we're in the process of testing in my lab right now. But one kind of easy experiment that we did early on was just to add an FC block to the co-cultures. So this is just the FC fragment of antibodies, which competes for the FC receptor. And we found that in the presence of FC blocking agent, we were able to reduce the percentage of killing and also the percentage of trogocytosis by about half. So it does seem that at least some of the trogocytic activity is due to antibodies and FC receptors. We're still not sure what the other half is due to, so these are things that we're currently testing. So the second question is, what enzymes are mediating this nibbling or this degradation of the parasite surface into these bites? And around this time, I was showing this data to someone who was at UCLA giving a talk, who was Victor Torres, that was a professor at NYU when I was there. And he said, I bet you it's serine proteases. Neutrophils have these 4 major serine proteases that have some properties that they can degrade bacteria. So I wonder if those proteases are being recruited into helping with these bites. And so we did a little reading about it. And in fact, the neutrophil serine proteases are required for killing bacteria in phagocytosis. And they've also been shown to be able to degrade bacterial membrane proteins and cause bacterial death. And it was kind of an easy experiment to do because they can be inhibited by commercially available serine protease inhibitors. So the next experiment that we did was to repeat our live imaging scheme where we're visualizing neutrophils trogocytosing the parasites. But we did it either in vehicle control conditions or after pre-incubating neutrophils with this serine protease inhibitor called AEBSF. And so you can see in the vehicle condition, we have accumulation of these bites of the parasite in the early minutes of the interaction. However, in the presence of this inhibitor, even though we still get some neutrophils homing to the parasite and surrounding, we never see any transfer of bites. So this does seem to inhibit trogocytosis. And again, we did this with many different donors and in many different videos, and we found that in the presence of this serine protease inhibitor, we always inhibited trogocytosis, so we inhibited the nibbling, and we also bring the levels of parasite killing down pretty low too. So it seems that the neutrophil serine proteases are involved in the trogocytosis and the killing of the parasites. Okay, so just as a recap, so we're about 40 minutes. So neutrophils can quickly and efficiently kill Trichomonas vaginalis in vitro. We found that the killing is strictly contact dependent and that bites of the parasite are taken prior to the death of the parasite, which is consistent with a death by trogocytosis or nibbling. That antibody FC receptor interactions and serine proteases are required for this to happen most efficiently. And this was, to our knowledge at the time, the first demonstration of neutrophils using trogocytosis to kill a pathogen. And so we think that trogocytosis could be a tool that neutrophils use for large motile threats. Okay, so around the same time, so our paper came out in 2018, and also in Cell in 2018, this paper came out showing that neutrophils kill antibody opsonized cancer cells using trogocytosis. And they use the word trogoptosis here, and they say that this means death following trogocytosis. So we could have used this word too, but the trogocytosis field is kind of enjoying a lot of different terminology. And so if you read reviews on the topic, you can read about all the different things that people are calling this really interesting cellular process where cells are kind of nibbling on each other. But this was a study of breast cancer cells that were coated with therapeutic monoclonal antibodies that are used against breast cancer, and showing that one of the ways that neutrophils can kill those cells is actually by nibbling on those cells until they die. So this was a huge collaboration between a lot of different investigators and clinicians. And they were able to do things that we were not able to do in our Trichomona system. And so I just want to briefly review this paper because I think it was a really great paper and really increases our confidence that this process is happening and that it's relevant in vivo. So one of the things they did was electron microscopy. So you can see these really high resolution pictures of the neutrophils taking nibbles of these cancer cells that are coated with the monoclonal antibodies. They were also able, because they were collaborating with clinicians, to get neutrophils from patients that were genetically deficient in the ability to degranulate. And so when they took neutrophils from these patients that could not degranulate, they showed that their neutrophils can still kill the cancer cells using trogocytosis. So again, this argues against this being a process that's mediated by neutrophil toxic granules, and that it really is a physical disruption of the membrane of the target cell. Again, the killing was potentiated by therapeutic monoclonal antibodies. So in a lot of instances of trogocytosis, in mammalian cells, we see that there's receptor ligand interactions that need to occur, and in a lot of cases, it's mediated by antibodies. And they showed this because when they blocked FC receptors, and so this is a complement receptor that binds IC3B, that complement opsonin. When they blocked these things, it abolished trogocytosis and killing. And another thing they did in this paper was they did intravital imaging in a mouse liver. And this is something that we were not able to do because there's no mouse model for Trichomonas. But in their mouse model, they're able to visualize these red tumor cells in the liver getting trogocytosed by neutrophils. So you can see the bites here. So this was really exciting because it really gave confidence to a lot of the work that we did with Trichomonas in vitro. It gave us more confidence that this process can happen in vivo. Although we haven't formally tested that yet because we don't have a mouse model. And so this really interesting cellular process of trogocytosis has been demonstrated to occur in many different circumstances across different kingdoms. So even protozoa were shown to be able to do this. So as I mentioned, the amoeba, Entamoeba histolytica, uses this nibbling process to kill host intestinal epithelial cells. White blood cells, neutrophils, and macrophages are shown to be doing this. Macrophages are shown to be doing this, and it's been shown as a way that intracellular bacteria can exploit this process to transfer from cell to cell while avoiding antibodies and other things that are out in the environment. This has been shown to be happening in the brain, so microglia can use this to remodel cells in the brain. And it's also been observed during development in C. elegans. And many people in this audience may know that trogocytosis occurs at the interface of T cells and APCs during antigen presentation. So I think that there's a lot more mixing of cell membrane proteins, and we can kind of challenge our definition about what the boundaries of a cell are. There seems to be a lot of instances of this one cell nibbling on adjacent cells going on throughout biology. Okay, so going back to our overarching question. So the first question, whether neutrophils can kill the parasite, and we found that the answer is yes, at least in vitro when we tested, and they do it using this process called trogocytosis. So our next question is, how does the parasite evade host immunity? And so another thing I haven't told you so far is that in all the experiments that I've shown you so far, we use a lab-adapted strain of the parasite, which is the standard convention in the field. That's the model that people use in most of their experiments. And so we wanted to know whether this happens more broadly with clinical isolates of Trichomonas. So we use our same cytolysis assay that I've been showing you, except now we use various clinical isolates of the parasite in the assay. And so when we did this, we actually found kind of a spectrum. So G3 is the lab-adapted strain that I've been showing you up until now. And so this is able to be killed by neutrophils. And these two other strains, LA1 and LSU160 that we have in the lab, are also able to be killed. But these two strains that actually come from the CDC, 1103 and 1132, those were kind of resistant to being killed by neutrophils. And so we think that while many strains can be killed, there may be some strains that cannot be killed by neutrophils. So they appear to exhibit some neutrophil evasive behavior. And we're not really certain of what the mechanisms of that are yet, but that's another thing that we're interested in looking into. But another thing that we wanted to ask was whether these strains that are kind of resistant to neutrophils are the more virulent strains. And so, so as I mentioned early on in the talk, one of the pathogenic behaviors of this parasite is to attach to vaginal epithelial cells and lyse them. And so as it turns out, we went back into the literature and checked out our two strains that were resistant to being killed by neutrophils, and found that these two strains are also really particularly lytic to host epithelial cells. And also in experiments that we did in 2016, where we challenged a whole panel of different kind of leukocytes with parasites, we found that the parasites were able to kill lymphocytes. So interestingly, they don't kill neutrophils or monocytes, but they are able to kill lymphocytes. And these strains that we think are more virulent because they kill epithelial cells more and they evade neutrophils were also more potent to kill lymphocytes. So it does seem that for all the in vitro tests that we have of characteristics of pathogenesis or virulence, that these two strains seem to be exhibiting those behaviors. And another interesting thing that I wanted to share, so as I mentioned, a lot of the strains of parasites that we have can actually kill lymphocytes. When we did this, when we co-culture the parasite with PBMCs, and then we stain to see who got killed, T cells or B cells, we find that the parasites actually show a preference for B cells. So there was a pretty substantial increase in the amount of B cell death compared to T cell death, even though everything was in the same well at the same time. So again, we think that these virulent strains of the parasites can kill lymphocytes, but they particularly seem to target B cells. And interestingly, old literature also showed that the parasites secrete cysteine proteases that degrade antibodies. So we think that these two in vitro properties support a model by which the parasite may be evading humoral immunity in vivo. And importantly, since we know that antibodies are involved in neutrophil killing the parasite by trogocytosis, if the parasite is degrading antibodies, if some of these more virulent strains have proteases that can degrade those antibodies, that could be one way that they're thwarting that process. Okay, so how does the parasite evade host immunity? So we think some virulent strains can evade being killed by neutrophils. And also they demonstrate properties, at least in vitro, that we think could promote evasion of humoral immunity. Okay, so we've got about 10 minutes left. So I just wanted to really briefly talk about pathogenesis. So just kind of the big picture. So how does this parasite cause disease? And so at Cal Poly, I teach a class called host pathogen interactions. And one of the main themes of the class is that disease is not only due to the microbe itself. So disease is caused by the microbe and due to the microbe, but it's also due to the host. So oftentimes the host response can be responsible for pathology, and of course other microbes that are present. And so in the case of trichomoniasis, we think that the disease is influenced by the parasite itself. So the parasite lysing those epithelial cells in the reproductive tract. But also the host response to that parasite. So we know neutrophils are recruited to the area. Sometimes they kill the parasite, but sometimes they don't. And in either case, we know that neutrophils are really inflammatory cells that can inflict collateral damage onto tissues. And the host microbiota is also going to play a role in this, because this is, at least in women, this is going on in the vagina, which is a rich microbial ecosystem. So the other microbes there can play a role. And in addition, we have to consider the parasite's microbiota. So this parasite actually oftentimes comes as a package deal. So the parasite has endosymbionts. So it houses a Mycoplasma species, and it also houses a virus called Trichomonas virus. So when you're thinking about immune responses to this parasite, you can think about immune responses that form against the Trichomona itself, but also against the Mycoplasma and also against the virus, if these symbionts are present in the parasite. So it's a little bit more likely to be a lot more complex in the context of natural infection. So I just wanted to briefly summarize some work of a colleague of mine in New Zealand, Augusto Barbosa. So he studies the interaction of Trichomonas with members of the vaginal microbiome. Vaginal microbiomes, a mark of health or eubiosis is actually lack of diversity. So it's kind of opposite of the gut microbiome, where we associate more diverse gut microbiomes with health. In the case of vaginal microbiomes, a state of eubiosis is generally a state where lactobacillus species are really dominating. And so Augusto actually found that lactobacillus can inhibit colonization of the parasite onto epithelial cell layers in vitro. So we think that lactobacilli in the vagina can have a protective effect against trichomoniasis. And interestingly, trichomoniasis cases are really strongly associated with kind of dysbiotic vaginal microbiomes. So vaginal microbiomes where there's outgrowth of anaerobic species, and particularly Gardnerella vaginalis, which is causative of bacterial vaginosis. So Trichomonas infection is strongly correlated with bacterial vaginosis. So we're not really sure what comes first, the chicken or the egg. So we're not sure if VV predisposes to trichomoniasis or whether the parasite goes in and wreaks havoc on the microbiome and causes this. Could be a little bit of both. But this is important to pay attention to because BV can also cause reproductive pathologies. And even in circumstances where trichomoniasis patients are cleared of the parasite using metronidazole, oftentimes the bacterial vaginosis state returns. And so trichomoniasis infection can be something that can cause a recurrent problem in the vaginal microbiome, independent of the parasite in the long term. And then regarding kind of this microbiome that the parasite has in itself, so 50% of the clinical isolates, and this really varies by geographic region. So in some places it's close to 100, and in some places it's around 10, but we say an average of 50% of the strains are positive for this double-stranded RNA virus that lives inside the parasite. And again, about half of the strains on average across geographic region test positive for this endosymbiont called Mycoplasma hominis, which also is inside the parasite. And so I wanted to mention this in this talk since this is associated with a CFAR. So for the strains that are positive for the virus, these strains can activate TLR3, which under normal circumstances the parasite does not activate. And this can cause the epithelial cells in the area to release more RANTES and CCL20, which can recruit more T cells to the area. So more targets for HIV potentially. So this is proposed to be one of the mechanisms that could link trichomoniasis with increased acquisition of HIV infection in patients. And interestingly, so a lot of this is the work of Reina Fikarova at Harvard. She also thinks that, so metronidazole treatment, which is the antibiotic that we use to treat trichomoniasis infection, so that causes lysis of the parasite. So she thinks that killing of the parasites with metronidazole causes those virions that are normally inside the parasite to be released out into the vaginal mucosa. And so this could actually enhance immunogenicity of the virus and kind of compound a lot of these TLR3 mediated mechanisms of recruiting more T cells to the area. And this Mycoplasma hominis symbiont is really interesting. So this is thought to activate TLR2. And another thing that I've looked at a lot in the lab is cytokine responses to the parasite. And it's a completely different picture whether or not the strain of Trichomonas that you're using has mycoplasma or not. And if the strain of Trichomonas that you're using has mycoplasma, you see thousands-fold higher IL-8 secretion. And IL-8 is the major neutrophil recruitment chemokine. And so we think that strains of the parasite that harbor this symbiont could be the ones that induce a lot of neutrophil recruitment to the area. And there's other inflammatory cytokines that are secreted in response to the parasite, like IL-6 and IL-1, that are really dependent on the presence of this symbiont. So if the strain does not harbor the symbiont, the parasites are kind of more immunologically silent and they don't really induce inflammatory cytokine. but if they harbor this symbiont, they induce a lot of it. So as you can imagine, we hypothesize that this really makes a huge difference in terms of inflammation. So important to note if the strains have these symbionts. And interestingly also, so this Mycoplasma hominis, some collaborators that we have in Italy recently identified that the mycoplasma actually produces a nuclease that can degrade neutrophil nets. So we don't think that netosis is involved in this really rapid killing that I showed you earlier on in my talk, but it's possible that in those strains that are evading that rapid killing, or in the context of a natural infection where there's biofilms and clumping and other things that may inhibit the neutrophils from killing using trogocytosis, it's possible that they can employ that netosis function later, and that this symbiont could be involved in conferring some resistance to neutrophils. So these are all things to think about in a natural infection that can complicate the scenario, and probably all things that contribute to pathogenesis and to a lot of the adverse sequelae that we see with infection. Okay, so overall conclusions are that neutrophils rapidly kill Trichomonas vaginalis using trogocytosis, that in vitro correlates of virulence are resistance to neutrophils and increased cytotoxicity towards urogenital epithelial cells and lymphocytes, and that in vivo pathogenesis is likely a result of the virulence of the individual parasite strain, but also whether or not that parasite harbors any symbionts. Host inflammation, which we think is mediated in large part by neutrophils, and also the host microbiome. So I can just leave up my acknowledgement slides and take any questions that folks may have.
Thank you, Francie. That was really great. I want to let other people, if they have any questions, chime in. I've got a small one, but anyone else, unmute yourself if you want. I'll start. You mentioned at the beginning that we know there's increasing rates of some drug resistance in Trichomonas, and I wonder if we know enough about the strains that seem to have the increased cytotoxicity, if they also correlate with increased drug resistance. One of the things we see in staph, for instance, is sometimes the most cytotoxic isn't always the most drug resistant, but other times, the scary cells, the scary strains do show up that have lots of cytotoxicity and drug resistance. Is that known for Trichomonas?
I'm not aware of any studies that have looked at a huge number of panels of strains and done that kind of analysis, but just anecdotally in the lab, no, I don't think we observe that, because we use dozens of different kind of strains in the Johnson lab, and yeah, the ones that are resistant are not necessarily the ones that are most lytic. So it doesn't seem like those things correlate.
That's good. I mean, slight reassurance.
Yeah. But
it's getting 2020 though, so I mean,
yeah,
it could still happen.
Yeah.
Hi, Mercy. This is Nadia. This was a very great presentation. I enjoyed it a lot.
Oh, good. Thanks for tuning in.
And I was just wondering whether any studies were done to see whether people who have the Trichomonas infection are actually more susceptible to HIV infection because lymphocytes are attracted to the area.
Yeah, so it seems, cohorts of sex workers in Africa, and I think there's been good, well-controlled studies with like 3 or 4 different groups, show that there's about a 3-fold increase in susceptibility of TB positive women to acquiring HIV. Interestingly, in cohorts of HIV positive women, the trichomoniasis, the rates of having active trichomoniasis is about 20%. So this is pretty high prevalence. So we think that the reasons are both due to the parasite having that lytic behavior toward the vaginal and cervical epithelial cells can increase the tissue damage and decrease the barrier function in that mucosa. That could, upon HIV encounter, increase the rate of virus passing through the mucosal tissues. But yeah, we also think that since there's leukocytes recruited to the Trichomonas infection, that brings more target cells that could be susceptible to HIV to the area. And so particularly in the Trichomonas strains that harbor that endosymbiont Trichomonas virus, we think that those are the strains that could disproportionately confer increased HIV susceptibility. So this, I don't think we have so trichomoniasis is not a reportable infection. So unfortunately, we don't have a ton of data about linking in vitro behaviors of virulence that we see in our experiments and linking that to what was going on in those patients and what their symptoms were like. So that's really unfortunate. But our prediction is that the increased HIV susceptibility is due both to tissue damage, recruitment of leukocytes to the area, and also that that's probably a bit more pronounced in the strains that harbor those symbionts.
So it probably would be possible to do some of these studies with people who come in for HIV infection, and they're tested for sexually transmitted diseases.
Yeah.
And they're found to be Trichomonas positive. Then maybe they can be sampled for whether they have the symbionts, what particular strains they have, so we could actually answer those questions.
Yeah, that would be a really cool study to do. Yeah.
Presumably it would be just in women, right? Or would there be a role in doing that in men? Just thinking about the epidemic a little bit in the United States.
So men are really largely asymptomatic, and the rates by which they self-resolve are higher. I don't know if it's more difficult to isolate trichomonads from men. It probably is. But in the data that I've seen with people that are seropositive for TB or people that have active infections of trichomoniasis, in terms of increasing susceptibility to HIV, I think that those links don't bear out in men. They only bear out in women. So
interesting. Okay. And
there's very, very little trichomonas transmission in men who have sex with men. So it's really transmitted during heterosexual sex, and it can be transmitted from vagina to vagina, but it doesn't get transmitted from male to male. So it's probably less common in a lot of the HIV cohorts in the US.
Here, but I mean, in other parts of the world, particularly sub-Saharan Africa, I mean, those cohorts are much more robust and could potentially do the study that Nadia is talking about.
Yeah.
I don't know how hard it is isolating the Trichomonas. Is it something, I guess there's two parts. One is we don't always detect HIV seroconversion for a few weeks, in even the best situation, might be months and more likely. And then the second one is how readily could a field clinic actually collect and isolate, TB?
Yeah. I wonder in men if it can be isolated from urine. I assume so, because the way that it would be tested for would be PCR test in urine. So there must be some full trichomonads that can make it down.
And I mean, we do know other. Oh, sorry. Other STIs will increase the rate at which men who are positive give HIV to negative partners, presumably through some inflammation in the male urogenital tract. And so there's potentially still value in understanding if there are HIV positive men who have TB and are therefore at higher risk of transmitting. But anyway. Good point, Nadia. Any other questions? I'm going to do a little applause here. Thank you so much for your presentation, Francie.
Thank you.
Come visit us sometime when this is all over. All right.
Okay.
Bye, everyone. Thanks.